Enzyme-substrate complex, a non-covalent complex that is composed of a substrate bonded to the active site of the enzyme. This is formed during a chemical reaction, and after the reaction, the enzyme can bond with another substrate to complete the complex again. Competitive inhibition. Competitive inhibition occurs when an inhibitor binds to the active site of the enzyme, which prevents the substrate from attaching and completing the reaction. The inhibitor may fit in the active site, but cannot be acted upon by the enzyme. Other active sites in the enzyme may still be viable because competitive inhibition does not alter the enzyme structure. Non-competitive inhibition. Non-competitive inhibition occurs when a molecule binds to a spot other than the active site on the enzyme. This inhibition changes the structure of the enzyme so the substrate can still bond to it, but the reaction will not be as efficient or as effective as the traditional complex. Allosteric regulation. Allosteric regulation is when a molecule binds to a site other than the active site and changes the behavior of the enzyme by adjusting its conformation. The allosteric molecule can increase or decrease the enzyme's activity. Feedback inhibition. Feedback inhibition refers to the mechanisms that control certain functions. Feedback inhibition is when a sensor gets a certain amount of feedback from its environment and turns another system on or off. Denaturing of an enzyme. When an enzyme is denatured, it can lose some of its original properties and may not be able to perform its original functions. Denaturing results in permanent change. However, enzymes never die since they are neither living or non-living.